Good morning, everyone. My name is Dr. Renee Binder. I am the director of the Forensic Psychiatry program at UCSF and Associate Dean in the School of Medicine. And this morning in Grand Rounds, we are in for a real treat. We will be hearing about what neuroscience can and cannot answer in the courtroom. And our speaker today is Dr. Octavio Choi. Dr. Choi is a clinical associate professor in the psychiatry department at Stanford University School of Medicine, and he specializes in neurolaw, which is an emerging interdisciplinary field that studies the use and misuse of neuroscience-based evidence in the courtroom. Dr. Choi is the founding director of Stanford's Forensic Psychiatry Fellowship, which is the first neurolaw-focused forensic fellowship in the world. And he's going to be having his first fellows in July of 2022. His research interests include the use of functional brain imaging to discern mental states such as lie detection and the neural basis for moral decision-making, such as how defects in moral reasoning brain centers may give rise to psychopathy. Dr. Choi graduated from Stanford University with a Bachelor of Science and a self-designed major of computational neuroscience. He received his MD and PhD at the University of California, San Diego, and actually he received a perfect score on his MCATs in 1992. Dr. Choi's PhD in neurosciences was awarded for research in molecular mechanisms of visual system development conducted at the Salk Institute. And after medical school, he completed his psych residency at UCLA and his fellowship in forensic psychiatry at the University of Pennsylvania. And just to remind everyone, participants can submit questions via chat. And if you'd like to ask a question live, you can click the reactions and raise your hand, and we will start the Q&A at about 20 after 9. So welcome, Dr. Choi.
Thank you so much for that very kind intro, Renee. I think I peaked at that MCAT. I should just stop right there. Okay, so let me just start my screen share. Well, it's an honor to be presenting Grand Rounds at UCSF. Thank you so much for the invitation. So yes, as Renee mentioned, I'm a forensic psychiatrist with a background in neuroscience, and I am a neurolaw specialist. So I'll just explain a little bit what that is. So neurolaw is what it sounds like. It's a study of neuroscience and the law. And one of the, this is from a recent review paper that shows the number of cases that invoke neuroscience is increasing year after year. And when you think about why that is, it's really because there's a great demand for what neuroscience may be able to offer. That's because mental states and capacities are of prime importance in the law. So we have the brain, and I kind of conceptualize capacities as a function of brain hardware, and of course the activity of the brain gives rise to mental states or the mind. So legally relevant brain capacities might be things like capacity for impulse control, capacity to think rationally, capacity for moral reasoning, and these are all capacities that are instantiated in various ways in the various insanity statutes. And just like an iPhone has chips and specialized hardware, and when you run, and the activity of those chips allows you to run apps, the hardware of our brain allows us to run apps, and those apps are mental states, things like lying, things like being in various amounts of pain, things like having a level of intent for your actions, and these are all mental states that are legally relevant. So, mental states are key to the law, and just focusing on the intent. You have a
bar on the side of your screen with the colors on it.
Oh, yeah, that's funny. Thank you for that. Yes, let me just get rid of these sidebars. Thanks for pointing that out, Renee. That's completely invisible to me. Can you see that now?
It's perfect now.
Okay, great. So, talking about mental states and focusing on the level of intent. So, many of you know this, many of you may not. So crimes are in general defined in two parts. One is it describes the forbidden act. So and that's called the actus reus. So in this case, the forbidden act might be killing a human being. But we're not done yet. So another really important part of this crime is the level of intent or the mental state. So that's called the mens rea. And so whether the mental state was accidental or intentional, that really determines how much we should punish people. That makes intuitive sense, right? And so the overall principle is that just punishment should take mental states into account. Now, pivoting to brain capacities. So brain capacities are also key to the law, things like moral reasoning, your ability to think rationally, your ability to control your impulses. And especially, not just that, but the law is very interested in how these capacities may change over time, how they may change with brain injuries and tumors and things like that. So brain capacities are key to the law, and the overall principle is that just punishment must take brain capacities into account. Both these prongs are incorporated in the Eighth Amendment's prohibition against cruel and unusual punishments. And the gist of it is that just punishment is proportional, that cruel and unusual punishments are disproportionate, so that just punishment has to be proportional, and proportional to the offense, and not just what kind of offense. Obviously, shoplifting is less punishment worthy than murder. So not just the act, but also the mental state, whether something was done intentionally or whether it was done accidentally is very important. But just punishment also is proportional to the offender, right? So this has to do with the brain capacities. If you're a baby, right, and you commit an offense that, you would blame that baby less than an adult. If someone's very elderly and suffering from dementia, you might, adjust your culpability because of that. So just punishment has to be proportional. It has to take into account both mental states, the amount of intent, as well as the brain capacities. Now, another reason why there's so much neuroscience in the courts is because there's so much neuroscience these days. We are in a golden age of neuroscience. And the first wave, is better and better ways to visualize brain structure. The second wave, are new tools to visualize brain activity. And the final wave are interventions where we can now focally neuromodulate. And you guys at UCSF have a wonderful interventional psychiatry team. I recently saw Catherine Scangos's grand rounds at Stanford a few weeks ago, and if you haven't seen that, it is mind-blowing. It's truly worth seeing. And of course, underlying all these advances in imaging are the advances in computing power, which kind of drive the whole revolution underneath. So I'm going to explore in this talk how much we can discern about brain capacities from looking at brain structure, how much we can discern about mental states by looking at brain activity. And I won't get to talk about intervention, but there's a wonderful suite of new treatments that are coming down the line that are well worth talking about in another talk. So the overview of the talk is I'm going to mention a few things about the basics of MRI. Then I'm going to go into some studies discerning mental states from brain activity, which is also called mind reading. We'll talk about a common kind of error that arises called reverse inference errors, and we'll talk about a landmark case called US v. Semrau. Then we'll move on to studies that demonstrate how you can discern capacities from brain structure. I'll go over a very interesting study about acquired criminality after brain injury and talk a little bit about the neural basis of moral reasoning. Finally, we'll go over some limitations of neuroevidence, and the big thing is really this G2I problem, and I'll discuss a landmark case called People v. Weinstein, and I'll finish up with my thoughts about what neuroevidence is good for. Okay, so neuroimaging, there's so many different modalities one can talk about. Because of time, I'm only going to focus on a few of them. I'm going to talk about MRI and its derivatives, DTI, fMRI, and resting state fMRI. So let's begin with MRI. So you guys are all familiar with MRIs, developed in the 70s. The people who developed it won a Nobel Prize in 2003. Now, the MRI is an incredibly complicated machine, and there are semester-long courses that talk about how the MRI works, and I'm going to discuss it in 1 minute. So the MRI, the key thing about the MRI machine is that it has 3 magnetic coils that set up 3 magnetic gradients 90 degrees from each other. So basically these 3 magnetic gradients set up an XYZ coordinate system that allows the machine to interrogate any spot in 3 dimensions of space and interrogate the magnetic qualities of any of these spots called voxels, which are like little boxes. They're like 3-dimensional pixels. So the magnets allow you to focus the interrogation to one spot in 3 dimensions of space, and interrogate the magnetic qualities there. And because different parts of the brain are made of different combinations of proteins and fats and water, they have slightly different magnetic qualities that can be discerned and translated into an image that we can recognize. We probably also know that magnetic field strength determines the MRI resolution, and as the magnets got stronger and stronger over time, the resolution increases. So when you look at the initial 0.35 Tesla magnets and compare that to the modern research 7 Tesla magnets, I mean, check out the cerebellum. The increase in resolution is just remarkable. This is the most powerful MRI currently in existence. It's a 10.5 Tesla magnet that's in Minnesota, and I just think it's interesting to appreciate how big these machines are getting because the magnets have to be so powerful these days. I'm looking forward to this. INUMAC, this is going to be the world's most powerful MRI. So that's a person that's in there. So you can see how big you have to make these machines to get the magnets so strong. It's going to generate an 11.75 Tesla field, which is even greater than the particle accelerators in Europe. And that increase in magnetic strength really gets you something. So it gets you a tenfold increase in resolution. So currently the 3 Tesla magnets can image about a 1 millimeter cube that contains 20,000 neurons. The 11.75 Tesla magnet will allow you to image cubes that have only 2,000 neurons. And like many large science projects, this is a consortium in Europe. It keeps being delayed, but 2022 is when they're saying it's finally going to produce some images. And really looking forward to what this machine can put out. So these kind of structural brain imaging studies have been a real boon for loss of function analyses. So if someone has a stroke and they can't move their left arm, we can immediately look at the scanner and see what part of their brain might have had the stroke and make those correlations. Oh, this part of the brain must be really important to control the left arm. That's a real acceleration from the old days when you only had invasive imaging. So in the old days, you had to, if someone came into your office looking like this, you'd have to wait until they die and then cut out their brain, slice it slice by slice to see what the defect is. So obviously being able to do this non-invasively in a living human has really vastly accelerated making these loss of function analyses. So loss of function studies are great, but they're quite limited. So the analogy would be like, it's sort of like trying to figure out how your iPhone works by taking an ice pick and stabbing random parts of it and seeing what apps don't run. So it's a pretty limited way to figure out how the brain works. What we really need is a way to non-invasively study normal brain function. And we had to wait until the advent of functional brain imaging to do this. So the 2 most common ways to visualize brain activity these days is with PET scans and fMRI. I'm going to mostly focus on fMRI. PET's a wonderful modality, but it's much less utilized because you have to inject radioactive tracers into people, and IRB boards don't like that. So fMRI is much more in use. Now, how does it work? So it's important to understand that fMRI does not work by directly imaging brain activity. It images a vascular response to brain activity. So what it's actually visualizing are relative oxygenation levels. And so it takes advantage of a phenomenon called neurovascular coupling. So this is basically the concept that when a local part of the brain is working hard, it recruits more blood flow. So here's the brain at rest, here's a blood vessel going through that brain at rest, and there's the normal amount of blood flow. But when this part of the brain is working hard, the blood vessels dilate, more blood rushes in, and these blue dots are oxygen. And because more blood is rushing in, there's actually a rise in oxygen level, which the machine can discern. So what's actually reading out is called the BOLD signal, blood oxygenation level dependent signal. And this is getting a little in the weeds, but I just find it so fascinating. You may wonder how the MRI is able to discern higher oxygen levels, and that's because when oxygen binds to iron, it changes the oxidation state, right? So oxygen binds to iron, and it actually rusts at a molecular level, which is why it turns a little bit more red. And that rusting at the molecular level, that change in oxidation state of the iron, also slightly changes the magnetic qualities, which the MRI can discern. So because of this magical quality of iron changing magnetic qualities when it's bound to oxygen, the MRI is able to tell you what parts of the brain have more oxygen and less oxygen. And parts of the brain that have more oxygen are, the theory is it's working harder, it's more active. Now, the key point is that all current functional imaging methods, not just fMRI, but all the others, all measure brain activity indirectly. So it visualizes a vascular response to brain activity, not brain activity itself. It's a highly processed image with many steps. And so with any correlations, there's lots of problems that can pop up. I think the biggest one legally to worry about is this phenomenon called dilated defect. This is the fact that there's no consistent set of standards to process an fMRI image. So by tweaking the settings in the many steps, you can make a defect look bigger or smaller. So this lack of standards is probably the biggest barrier, I would say, for admission of fMRI evidence into the courtroom. Nevertheless, fMRI is sort of the best that we have, and it is amazingly good for what it does. And so this is just a standard fMRI image of someone hearing voices versus seeing pictures, activating auditory cortex versus visual cortex. And structural imaging studies, functional imaging studies, and now ability to locally neuromodulate have all converged into the principle of functional specialization, which is basically the simple idea that different areas of the brain do different things. This is something that we take for granted these days, but it was not taken for granted 50, 60, 70 years ago. Okay, so that's all I'm going to say about MRI. Now let's look at some applications of fMRI, try to discern mental states from brain activity. Okay, so so the brain has activity generating the mental state, and so one of the basic questions you might have is it even possible to mind read from brain activity? And in my opinion, the answer is yes. In theory, if distinct mental states are encoded by distinct brain states, which most neuroscientists think is true. So your brain has a certain amount of activity, gives rise to your mental state, your brain changes activity, has a different mental state, different activity, different mental state. So as long as different kinds of brain activity give rise to different kinds of mental states, we should be able to do the reverse where we look at the mental state and guess we should be able to look at brain activity and guess what mental state someone's in. So this is the first brain reading study. This was back in 2000 with Nancy Conwisher at MIT. So Nancy spent much of the 90s interested in two specialized areas of the brain, one called the FFA and the other called the PPA. So FFA is an area that she was remarkable because it gets very active and happy when you show the person pictures of faces. So she called this area the fusiform face area. The PPA seems to preferentially activate when seeing images of places, landmarks, buildings, things like that. So Nancy called this area of the cortex PPA, or parahippocampal place area. And so this is an example of functional specialization. This seems to be a face selective area. This seems to be a place selective area. And so she did the simple experiment of showing people in the scanner either a face or a place, and the researchers would simply inspect what parts of the brain were lighting up and guess, just looking at the activity of these two different places, whether the subject was looking at a face or a place, and they can guess with 85% accuracy. So modest beginnings, but it's a proof of principle that one is able to, at a basic level, make some good guesses about what someone might be looking at. Here's an interesting clinical application of this. So there's a bunch of people in vegetative states or minimally responsive states in the hospital. And the question has always been, are these people actually in a deep coma and unaware, or are they actually aware but locked in so they can't respond? And previous to fMRI, it was really hard to answer this question. So, if you show a normal person, a control subject, and put him in the scanner and say, imagine playing tennis, various parts of the brain light up, including the supplemental motor area. If you then tell the subject to imagine walking around your house, a different subset of areas light up, including the parahippocampal place area. So they wheeled one of the patients in this minimally responsive state also into the scanner and told her, imagine playing tennis, and the supplemental motor cortex lit up. And then they told her to imagine walking around her house, and the correct areas for consistent with the controls lit up. So she was able to willfully modulate her brain activity depending on what the researchers were telling her to do. So they concluded that she was actually aware but locked in. So they kept her, they didn't unplug her, and she eventually woke up. They replicated the study a few years later on 54 subjects. This is the study that was published in a New England Journal. And out of 54 hospitalized patients in this minimally responsive state, they were able to identify 5 able to willfully modulate their brain activity. This is kind of just a very interesting application of mind reading to determine who we should keep alive. So shifting to lying, is it possible to look at someone's brain activity and discern whether they're lying or not? So before scanners came along, psychologists have been studying lying for a long time, and they came up with this 3-factor model, basically saying, lying is difficult. There's a bunch of things you need to do right to lie well. There's a bunch of cognitive things you have to do. You have to inhibit the truth. This is assuming that the default communication mode is telling the truth. So you have to suppress the natural default mode. Then you have to generate a plausible alternative. You need to keep your story straight so memory systems are active. There might be an emotional component. Many people, when they're lying, feel more anxious or guilty or excited, and that has to be suppressed with acting. You have to manage your emotions. You have to inhibit lying behaviors. You have to generate honest behaviors, like looking people right in the eye. So it's quite difficult, which is why one of the first truth-compelling technologies was wine, right? Because it just makes it a lot harder to do all the acting and cognitive tasks you need to lie well. So this is the first neural correlates of deception study. Spence in 2001 put people on a scanner and had them tell the truth, had them lie, had them tell the truth, tell them lie. And he compared the brain activation maps of people telling the truth versus lying. And this is what he reported. So he was able to identify a few distinct areas of the brain that preferentially lit up when the person was lying versus telling the truth. And these are areas that make sense. So this one area here is called the ACC, which many of you will recognize as one of the brain's conflict detectors. And so the interpretation of this activity was that the default mode, your normal goal for communication is to tell the truth, but in this case you're going to tell a lie. So there's a conflict of goals. So the ACC becomes active to resolve the conflict. Another area of the brain that lit up was the ventrolateral prefrontal cortex, which is an area of the brain involved in response inhibition. So the idea is, you want to tell the truth, but you're not going to tell the truth. So you have to suppress that, and that requires some brain activity. So it's like, when you're at Thanksgiving and you have that kind of the uncle that's spouting off conspiracy theories, and you just need to hold your tongue, and it's a really hard effort, right? That effort is instantiated by ventrolateral prefrontal cortex activity telling you, don't talk, just stay quiet. Now, over time, more and more brain areas were discovered. They make sense. So there are things like the ventrolateral prefrontal cortex that we talked about and the ACC that we talked about. But the dorsolateral prefrontal cortex becomes active when you're working hard, generating novel solutions, coming up with a new story. The ventromedial prefrontal cortex is involved in emotional regulation to kind of inhibit signs of anxiety. So these all make sense. And so, so then the next natural step would be, well, can we just like Nancy Conwisher did with the face selective areas, can we just look at these few spots of the brain and make a guess about whether someone's lying or not? So that was done in 2005, and it's called the region of interest approach. We take a few little spots. It's mostly visual inspection. And the long story short is they're between 76 and 78% accurate as a lie detector in this very controlled laboratory study. So not bad, right? Now, but the problem when you just look at a couple of spots and you infer a mental state from the brain activity of a few spots are reverse inference errors. So let me describe what this is. So I've been involved in a bunch of cases which basically summed up as my amygdala made me do it. And so, this is taken from an actual, case that I was working on. And basically, the opposing expert was saying, well, look at Mr. B's amygdala. And I'm not even going to describe what this is, but let's just take it for granted that this means that the amygdala is overactive. He says figure 7 shows that at rest, Mr. B's amygdala is red hot. Thus, Mr. B is hypervigilant, alert for and sensing danger in the environment. And then he goes on to say the amygdala is a vitally important structure in the brain. There is, in fact, a Wikipedia page devoted solely to the structure. Then he quotes 2 pages from Wikipedia. The gist of it is there is a direct relationship between the activation of the amygdala and the level of anxieties the subject feels. And so, the expert was using this kind of amygdala overactivity to make the case this is a person who killed another person in the heat of passion. So to showing that his amygdala is too active, he's anxious all the time, and that's why he should be mitigated in terms of his acts. Now, what's the problem with this line of reasoning? So the amygdala is a deep structure in the temporal lobe, right? It sits right there, right? And there, it is true that many studies show that the amygdala responds to fearful states. So when this is a classic experiment where you show subjects images of a neutral face versus a fearful face, and when you compare the brain activity between these two, you can identify the amygdala as lighting up. So this has been the basis of what is now the kind of folk wisdom that the amygdala is the fear center of the brain. So there's lots of studies that establish that if you put someone in a state of fear by showing them like a scary picture, that their amygdala would light up. This is a strong inference. This is supported by many different studies. But the reverse inference is just by looking at amygdala activity, can you make the reverse inference that person was necessarily in a state of fear? What else would you have to know to determine how strong this reverse inference is? And in this setting, I'm just asking a rhetorical question. If we're all in class, I'd be expecting answers. But the basic gist of it is, you have to understand what other things make the amygdala light up. And the more other things that make the amygdala light up, the weaker this particular reverse inference becomes, right? So let me give you some examples of other things that make the amygdala light up. Donuts, if you're hungry. Semi-nudes. Weird, novel things like chrome rhinoceri. George Clooney, if you're into George Clooney. Mother Teresa, if you're into Mother Teresa. So Because there are so many other things that make the amygdala light up. Just making, you can't just say, well, the activity is, the amygdala is lighting up, therefore the person's in a state of fear. So, the overall gist of, the modern understanding of what the amygdala is preferentially selected for is salience. So the amygdala lights up when it detects things in the environment it thinks you might be interested in knowing about. So of course, things that are threats to you are salient stimuli because your survival is at stake. But if you're hungry, donuts are salient stimuli, which will make your amygdala light up. George Clooney, if you're into George Clooney, will make your amygdala light up. So, amygdala activates a salience, not just to fear. Right, and these are actually actual examples taken from high quality. They weren't taken from Wikipedia; they're taken from actual high quality studies. And I'm happy to share these slides with anyone who wants them. I try to put the references in each slide that back up what I'm saying. So, many current experts in the court are stuck in phrenological thinking. So phrenological thinking was like there's a one-to-one mapping of brain area to function. And so phrenology obviously has many problems. One of the first problems is that these brain areas don't seem to be related to reality. So why is this the imitation part of the brain? Why is this the veneration part of the brain? Right, so it's unclear to me how these brain areas arose. But the second error, is that, they have a very simplistic understanding of how the brain works, where, each area of the brain does one thing, and activity of that one area of the brain leads to one function. The simplistic one-to-one mapping of brain area to function is simply incorrect, and I want everyone to take home that message. So the idea is, it got part of it right. The brain areas are specialized, right? So they're specialized in terms of vision and language and motor control, but each brain area is actually activated by multiple tasks and mental states. So the activity of a single brain area has multiple interpretations, making that reverse inference quite weak. So the analogy, again, is like your iPhone, right? So your iPhone has chips on it, but there's not a one-to-one correspondence between chips and apps, right? There's no Angry Birds chip on your iPhone. There's no Instagram chip on your iPhone, right? There are chips to process the camera and graphics and sound, and those chips all work in concert, right, to run any particular app. Similarly, our brain has areas for memory processing, for sound and vision, and depending on the task that we're running, it's going to activate different subsets of these specialized modules. So this is a concept called distributed processing, right? So the brain areas are specialized, but not to the extreme where there's a one-to-one mapping of brain area to function. Now, so actually, brain states like deception actually activate entire brain-wide patterns of brain activity. And so to really accurately get at mind reading from brain activity, you have to actually look at the entire brain's activity pattern. That's too complicated for people. So how can we do this? We use our robot friends. So the advent of AI has been a real revolution in cracking mind reading. So here's how you build a mind reader. You put someone in a scanner, and you manipulate their mental state. Make them lie, make them tell the truth, make them lie, make them tell the truth. Manipulate the level of pain that they're in, manipulate what level of intent that they have, and you study their corresponding brain activity. So modern AI systems learn by showing, so then you train an AI. You say to the AI, this is the mental state, here's the brain activity. Here's the mental state, here's the brain activity. You show it lots and lots of examples of different mental states correlated with the brain activity. And at a certain point, the AI is trained, and you can just give it the brain activity and make it guess what the mental state is. So this has actually been done. So this is a paper that really, rocked a lot of people, reconstructing visual experiences from brain activity evoked by natural movies. This is Jack Gallant's lab at UC Berkeley. He's at the forefront of mind reading from brain activity. So this is a study where they put someone in the scanner and they play them a YouTube clip. So you can see it running in the monitor here, and you can see it blown up here on the left. At the same time, they're reading out brain activity, and they're training the AI. Here's the video clip, here's the brain activity. Here's the video clip, here's the brain activity. Over and over again, lots of examples of video clip and brain activity. And once the AI is sufficiently trained, you could just show it the brain activity and say, can you guess what they're looking at? So what we're seeing on the right, so on the left is what the subject is actually seeing. On the right is what the computer has reconstructed, is guessing what the person is seeing just by looking at their brain activity. So it's kind of blurry, it's kind of low resolution, but the fact is it can be done, and it obviously needs to be refined. But this paper back in 2011 was really quite a breakthrough, showing how far you can get in guessing what someone's looking at just by looking at their brain activity. I thought just for this crowd you might find this interesting. People are also using the same technology to try to visualize dream imagery. So, this was back in 2013. This paper wasn't able to do it, but wouldn't it be cool as psychiatrists if we could just say, instead of saying, what did you dream about last night? If we can just say like, oh, let's look at the tape. That day is coming. As applied to lie detection, so people have used these AI methods where they're looking at the entire brain activation patterns. And to make a long story short, they are between 90 and 99% accurate in these small studies when you start to use AI. So that raises a lot of questions, right? So is this ready for the court? And so US v. Semrau was the first attempt to introduce fMRI-based lie detection into a court case. So Semrau was a doctor in Tennessee who was accused of Medicare, Medicaid fraud. The mens rea for fraud is things have to be done knowingly. You have to act with an intent to defraud. And his defense was basically, this is an honest mistake. I had no intent to defraud. So he hired a private fMRI lie detection company that no longer exists to basically back up his claims that this was all an honest mistake. And you might not know this, but you can't just introduce any scientific evidence you want in the trial phase of a criminal trial. It has to pass through certain standards. The trial judge has discretion about what to allow in and what to exclude. And so they often hold pre-trial hearings to determine that. So they're called Daubert hearings. Right, so these Daubert hearings, basically the judge is looking to see whether the science you're trying to introduce in the trial case is good science, and the various Daubert factors are things like testability and peer review and error rates. Do standards exist, and is the method generally accepted? I'm just going to focus on, and so it failed a bunch of these things. So the judge, in my opinion, properly excluded this evidence from the trial. But I want to just talk about error rates. That's, I think, the most salient take-home message. Basically, they were claiming, the person who ran the private fMRI company saying, well, my thing is, my lie detector is 94% accurate. And the judge basically said, well, that might be true or not, but basically the sample size is too small. There are no known error rates for fMRI-based lie detection outside the lab setting. So the error rate of real-life fMRI-based lie detection is unknown. So this is a problem of ecological validity. And this is a problem that plagues most neuroscience efforts to be relevant in the courtroom. So ecological validity, many of us know this already, but it's basically how relevant is a study to the patients I treat, right? So there's a population to study and the population of interest, which might be like my patients, right? And so the more if the population of the study overlaps highly with my patient's population, then the study is highly relevant. You might say, oh, I might want to try that medication on my patients. But of course, if the population of the study and your own patients are highly divergent, then the study is less relevant. So the judge's conclusion is basically lying in the lab is very different from lying in the court. In studies, it's different populations, and it's also a different task. So people who in lie detection studies are generally young male undergraduates who are right-handed. They're highly educated, they have no mental illness and so forth, whereas criminal defendants might be an older population, more subject to mental illness and traumatic brain injury and so forth. Also, the tasks are very different. Lying, as compliance to follow the researcher's instructions in a scanner is very different than lying in a court situation where you're lying as defiance and you can incorporate countermeasures and all sorts of things. So for all these reasons, the lie detection studies were deemed not ecologically valid. Not the real world, so it lacks ecological validity. And basically, this problem with ecological validity applies to all attempts to decode mental states from brain activity. So attempts to discern how much pain someone's in, attempts to read people's memories out. They're all done on small studies in non-representative populations doing non-representative tasks. So they tend to create underpowered and skewed AIs, but they establish a proof of principle. However, it's insufficiently tested in real world situations. So the basic conclusion is that mind reading from brain activity is not yet ready for the courtroom. Of course, one of the most exciting things about neuroscience is that it's always progressing. So I'm definitely prepared to update my opinion as more data comes in. Moving along to discerning capacities from brain structure. So again, one of the things I didn't mention in this slide, with all these new technologies, is another trend that's really very, very relevant, which is the rise of collaborative neuroscience. So this is basically labs getting together and collaborating, pooling their studies, right? And a lot of this was driven by the BRAIN Initiative, which is a big NIMH initiative to fund these kind of collaborative projects. One of these large projects is the Human Connectome projects, which has really been a revolution. So before the Human Connectome Project, we were getting a better idea of what different brain areas did, but we didn't know how they were connected. What the Human Connectome Project is doing is allowing us to finally see the underlying blueprint or the circuit diagram of the brain. And it's doing this by two novel imaging methods called DTI and resting state fMRI. Before these technologies came along, it was really hard to determine axonal pathways. Typically, you had to do it in lab animals where you inject a fluorescent dye and let it diffuse over time, then kill the animal, slice up the brain, and see the fluorescent pathways lighting up. Obviously, you can't do that in a living human, right? But with DTI and resting state fMRI, you can. So DTI allows you to generate maps of anatomical connectivity, and resting state fMRI allows you to generate maps of functional connectivity. Just very briefly, the way DTI works is that MRIs actually do not have the resolution to visualize axons, but it can discern the movement, overall diffusion of water molecules. And because water molecules are more likely to diffuse up and down the axon rather than across it, just by looking at where the water molecules are going, you can discern where the axonal pathways are. So it's like if you were looking from above at roads and cars, and you can't see the roads, but just by looking at how the cars are moving, you can discern where the roads are. So DTI gives you a map of anatomical connectivity. Resting state fMRI answers the question of how brain areas are connected. So task-based fMRI are like what areas of brain do what. So in this case, someone's moving their right hand and the left motor cortex is lighting up. Resting state fMRI, the person's not doing any particular task. They're just kind of daydreaming in the scanner, and there are natural fluctuations in brain activity that just occur when you're daydreaming. And so what resting state fMRI is doing is looking at what areas of the brain activity is correlated, right? Because if they're correlated, they must be connected in somehow. They must be functionally connected in some way. So these two techniques, DTI and resting state fMRI, have allowed us to establish a very good normative database for the brain circuitry, right? So far they've scanned 1,100 healthy subjects with standardized high-resolution protocols, and they've made all that data publicly accessible in a connectome database, which is really leading to a lot of new insights. I'll give you an example. This paper on acquired criminality. So this is a paper published in 2018 where they they did a big literature search to identify people who suffered a brain injury and then had new onset criminality soon after that. They identified 17 cases in the literature, so here are the 17 brains, and the red parts are where the lesion was. So these are 17 brains who had no criminal history, had some kind of brain lesion, and then soon thereafter started to commit crimes. And one of the observations you might have is, well, they're kind of just all over the place, so not all that interesting. But they asked the next interesting question, because now we have the connectome database. They asked what areas of these brain areas connected to. So they took brain number 1, here's the lesion, and they interrogated the connectome database, what are the brain areas that are connected to this area number 1? And they get a functional connectivity map. They do the same thing with brain number 2, functional connectivity map, and so forth with all the brains. You get a bunch of functional connectivity maps, and they observed that actually all these maps were basically the same map. There was a very high degree of network overlap, and they called this the criminality-associated network. Now, the basic kind of story of this is that, so basically all the 17 brains who had brain lesions and committed crimes had defects somewhere in this criminality-associated network. And they asked the next really interesting question, which is, what is this network? Do we know what this network is? They did a bit of literature search and said, yes, we know what this network is. This is actually the moral reasoning network. So the theory is that, that defects in any part of this moral reasoning network will lead to aberrant moral reasoning and therefore increase your risks of committing crimes. One of the key nodes of the moral reasoning network is the ventromedial prefrontal cortex. And let me talk about this for a little bit. So there's been a lot of studies that have converged to indicate that this ventromedial prefrontal cortex is one of the key moral reasoning centers in the brain. So there's the TBI literature, like the acquired criminality study I just mentioned. There are moral reasoning studies where you put people in scanners while cracking moral reasoning dilemmas, and this area of the brain becomes very active. There's a large literature on psychopaths and how psychopaths have underactive and atrophied ventromedial prefrontal cortices. And there's a rich dementia literature, things like frontotemporal dementia, which can have degeneration of this part of the brain, leading to aberrant behaviors. So when you think about, and I'm not going to have time to go into this, but the ventromedial cortex has lots of circuitry passing through it. I believe 3 essential circuits, that play a role in moral reasoning are punishment learning, impulse control, and empathy. And so if any of these kind of functions are taken offline, you're more likely to harm other people and to commit crimes. You can catch one of my psychopathy talks online if you want to see a deep dive of this. Of course, we know about Phineas Gage, who had an injury to his ventral medial prefrontal cortex, and soon after that injury started to act without social inhibitions, violating social norms and so forth. This is a very interesting case called Mr. Oft in the literature, where someone had new onset pedophilic behaviors and was later found to have a big tumor in his ventral medial prefrontal cortex, which was then removed, which led to resolution of pedophilic behaviors. Then his pedophilic behaviors came back, and when he looked back, his tumor had come back. So this is a very interesting case to discuss perhaps in the questions. The ventral medial prefrontal cortex, and more largely the OFC, is a very susceptible area because it resides on the undersurface of the brain, right? Which is a key area that gets injured when you hit your head on a steering wheel, for instance. And the ventral medial prefrontal cortex sits on top of the cribriform plate, so it's like cheese on a cheese grater. So this is a very easy area of the brain to damage, and I've had a bunch of cases involving OFC damage. Okay, let's just briefly go into the limitations of neuroevidence. Now, in general, there are 3 hurdles that neuroevidence has to pass to be useful in the courtroom, and neuroscience has trouble clearing each of these hurdles. So the first hurdle is simply establishing, is the finding abnormal? The second hurdle is, if you establish that the finding is abnormal, does the abnormality cause impairment? And finally, if it's abnormal and if it causes impairment, is it even relevant to the legal question? Now, it's surprisingly difficult for neuroscience to establish whether a finding is even abnormal, and that's because we lack normative databases. So what is a normative database? It's something that allows us to establish whether any given finding is abnormal or normal. So as a thought experiment, imagine you were a Martian, right? And you came down to Earth and you want to know, what do normal people look like? So let's say they sample these 9 people and say, okay, I've trained my neural net, I've looked at 9 people, now I know what normal people look like. But then what happens when you come across this guy? Abnormal or normal, right? For the Martian, it's say abnormal because their normative database is not big enough. Of course, we as people have encountered lots of people, so we know we have a large normative database. What about this thing? Highly abnormal, according to the Martian. This is actually my baby Dean. But we, of course, know this is a perfectly normal baby. What about teens? What about older people? What about people with face tattoos? These are all things that the Martian would classify as abnormal simply because the normative database isn't big enough. So basically, to answer the question of whether some phenomenon is normal or abnormal, you have to have a very large normative database. There's lots more I can say about this. But basically, in general, for most neural studies, we need larger samples and ecologically relevant samples. And in general, neural-based tests lack these kind of adequate normative databases. And without these databases, we can't even pass the first hurdle of establishing whether a finding is normal or abnormal. So moving on to the second, if abnormal, does abnormality cause impairment? And this is a real big problem. This is a big hurdle. It's a hard hurdle to pass because of biovariability. And I'm going to just illustrate it with a case called People v. Weinstein. So Herbert Weinstein was a 65-year-old ad executive with no prior criminal history. And he was, by all accounts, an upstanding citizen. But he got into an argument with his second wife, then threw her out the 12-story window, and she died, then rearranged things to make it look like a suicide. And we know this because he eventually confessed. Now, his defense team thought this was so odd that they got a brain scan, and this is what they found. A large arachnoid cyst with, this is the PET scan showing no metabolic activity, and here's a structural MRI. Of course, you can imagine the defense team went, woohoo, we have our case. And they were planning to introduce these neuroimages as evidence that Herbert Weinstein was insane. So as you can't just introduce any kind of evidence you want. You have to pass, certain admissibility standards. And so a pretrial Fry hearing was held in New York, and the judge actually decided to allow the brain images into the trial phase of the criminal trial. At that point, the DA kind of lost their nerve, and they agreed to a plea bargain, letting Weinstein plead guilty to a lesser charge of manslaughter. And the defense counsel said, the prosecutor would never have agreed to a plea if the judge had excluded the PET evidence. So, biovariability is a really big problem. Because, the real question of Herbert Weinstein is, did his brain defect cause impairment? I mean, that's just the basic question, right? So that's a really hard question to answer, actually. Here's an easier question to answer that we still can't answer, which is just in general, not for Herbert Weinstein, but in general, for people who have this kind of defect, what percentage of them have functional impairment? And surprisingly, we do not have the answer to this question. because people often refer to studies that like this is a made-up study, but it's like many other studies where this is like 10 healthy people, 10 people frontal lobe damage. You measure impulse control, and on average, healthy people have greater impulse control than people with frontal lobe damage. Let's say. Now, does this study by itself tell you the likelihood of impairment from frontal lobe damage? The answer is no, because these are just average statistics, right? It doesn't give you much information about this, about the spread. So it doesn't give you the percentage likelihood that any given individual with frontal lobe damage will have an impulse control problem. Now, let's say some studies actually report subject by subject all the amount of impairment. So let's say the study did that, and it reported that 9 out of 10 of these people with frontal lobe damage had impairment in impulse control. Does that mean that for Mr. Weinstein that there was a 90% chance that, can we just extrapolate the study to say that Mr. Weinstein had a 90% chance of being impaired from his tumor? And again, if we had more time, I'd kind of like to soak in, but the answer is no. So here's a study, right, showing, let's say they're saying 9 out of 10 people with frontal lobe damage had some sort of impairment. And the question is, can we extrapolate this to, saying, does it mean 90% of people in general are impaired with this brain defect? And so you really have to notice is how the subjects got into the study. And so in this case, it's a neurology clinic, right? It's a skewed sample. None of you got up this morning and said, oh, I'm feeling great, it's time to get a brain scan. These are all symptomatic people that went to see their doctor who then got the brain scan. So in this skewed sample, 9 out of 10 were impaired. But Mr. Weinstein was not part of a neurology clinic, right? What we really want to know is, in the general population, what percentage of people with this defect are impaired? And to answer that question is really hard because, first of all, you have to scan a ton of people. It's super expensive because most people's brains will not have defects. So you have to scan enough people just to find enough that have the defect, right, which is shown in blue here. Then once you do that, so in this case, let's say there's 5 people in the sample, right, that have this defect, and you have to, so sorry, 5 people with a defect, and then you have to do some kind of functional test. How many of these 5 have impairment? Let's say 1 out of 5 have impairment. Then we have the answer, right? This means that in this study, 20% of the people have impairment, right? But we don't, since the study hasn't been done, we don't know whether this number is 20% or 80%, or anything in between. We simply do not know. So in general, the take-home message is that we lack large prospective studies of brain defects in the general population correlated with impairment. There's a lack of normative databases, right? And so we simply cannot answer this question. To give you a real-life example from CTE, this is a paper from 2017. I'm being mindful of the time here, but we are wrapping up. 2017, they basically looked at NFL football players, and they took 111 NFL players and found CTE in 110 of them. So 99% prevalence. So does this mean that NFL football players have a 99% chance of having CTE? This was a study that was invoked by the opposing expert in a CTE trial that I was involved in. You'd be forgiven if you thought that, because that's what the media certainly reported. And these are all from high-quality sources like CNN and NPR and New York Times. CTE found in 99% of studied brains from deceased NFL players. CTE found in 99% of former NFL players studied. You'd be forgiving for just assuming that this means 99% of NFL players' brains have CTE. But the real question to ask is, how did they get their sample, right? So when you really dive down into the study, really the key is, how do people get into the study? And they go deep, buried in the text is, the limitation is ascertainment bias, and you can't, because, this was a voluntary, caution has to be used in interpreting the high frequency of CTE. Estimates of prevalence cannot be concluded or implied. And actually, she's saying that the actual prevalence is unknown. And when I looked at an interview with Dr. McKee, who's the lead, the senior author of the study, she basically said there's lots of limitations. This is a brain donation study. It's a highly skewed population. The brains were donated by family members who in almost all cases were concerned about their loved ones. So this is not just a general, sampling of generally NFL players. These are NFL players that were having a lot of symptoms that got their family concerned. So you can't just extrapolate these small skewed samples to the more general population. So I know what you're saying. Some of you are saying, come on, can this guy really be unimpaired? The answer is yes. There haven't been a whole lot of general imaging studies in the general population, but there have been some. This is one from Rotterdam. They basically found 10% of just people off the street having significant brain findings. And here's some examples of significant brain findings that they found. And these are all walking, talking, grossly asymptomatic people. So you can have large defects and be asymptomatic. So this is the heart of the G2I problem, which is basically because of biovariability, research studies demonstrate differences based on groups, courts make decisions based on individuals, researchers generalize, and courts particularize. Biovariability makes it really hard to predict impairment in individuals with brain defects. And what are the sources of biovariabilities? There's many different sources, but I want to focus on neuroplasticity, the fact that their brains rewire over time. Right, this has been studied a lot in musicians and meditators and liars. Here's an example of 13 children who were born with extensive brain damage and perinatal brain damage, and they were scanned from the ages 7 to 13 years later and given IQ testing. So let's play this game called Guess My IQ. So this is a kid whose brain was heavily damaged around the time of birth, right? Verbal IQ 106, performance IQ 100, full scale IQ 104. Guess my IQ, right? Full scale IQ 99. Lots of studies. Here's a study of all high functioning adults who were missing half their brain due to childhood hemispherectomy. And they all had normal brain functioning. And this whole study was like looking at the rewiring of these half brain people, but they're all high functioning adults. You can have half a brain and be high functioning. And that's because of neuroplasticity. I'm not going to go into a last prong because of time, but basically, the cases I get involved in, and we're wrapping up here, Renee. So, is that I look at evidence basically from 3 different sources. There's usually some kind of neuro exam, neuroimaging, neuropsych testing, and behaviors. And when all these 3 streams of evidence converge, then you can get a really strong case. And so neuro can be really useful in these kind of cases, but often these things diverge. So what do you do then? When these different things conflict, the way I personally rank these behaviors and these evidences in reliability is I place the most reliability on behaviors. Just the classic thing, the behaviors around the time of the incident, behaviors in non-forensic contexts. I've really spent a lot of time finding temporal correlations of when the brain injury happened and when the behaviors occurred. Right? Neuropsych testing is much more reliable than neuroimaging. It's been tested, sensitivity and specificity tested, and norm testing has all been done. And neuroimaging is the least reliable because of all the problems I mentioned before. So the mantra is bad-looking brains can function well and good-looking brains can function badly. So the way I view neuroimaging is that it is a hypothesis generator, not a confirmatory test. So a neuro finding will certainly raise interesting questions, but whether that neuro finding actually has legal relevance depends on their behaviors and neuropsych testing, which in my mind are the confirmatory tests. So in the case of Herbert Weinstein, the evidence did conflict. The behavioral evidence showed no defects in his behaviors. Looking at his financial records and his personal journals, there were no discernible defects in his behaviors. But the neuroimaging obviously showed a big defect. So in this case, I would personally privilege the behavioral evidence over the neuroimaging. I believe the DEA caved a little bit too soon. Okay, so finally to the last few slides, what is neuroevidence good for? Hopefully I've impressed upon you in the first part of the talk that neuroscience is amazing. And that amazingness of neuroscience gives it a seductive allure that in the wrong hands of so-called experts can really do a lot of harm. We need to be humble. So this is an imaging study of using reconstruction of EM, electron micrographs, imaging a 3-dimensional reconstruction of 1/800 billionth of the brain. And the big take-home message is that the brain is incredibly complicated. This is 1/800 billionth of the brain, and it is packed with neurocircuitry. I can show you lots of diagrams to demonstrate that the brain is complex, but I'm sure you get it. So finally, what is neuroscience good for? Again, when it converges with other types of evidence, I find it very useful. In general, it's less suitable for the trial phase because of its higher standards, and it's more suitable, in my opinion, for mitigation and diversion, which tend to have looser standards. So sentencing, you can introduce mitigating factors. Pre-trial, you can do things like argue for a mental health diversion, which I'm a big fan of. Many of us know this. In the last 50 years, there's been an inversion of the seriously mentally ill used to be in hospitals, and now they're being treated in prisons, right? So there is something really amiss going on. Mental health diversion allows some people with serious mental illnesses, whose mental illness was a significant factor in the commission of the crime, to divert them out of the criminal justice pathway and into a mental health court where they can be treated properly. So neuroinferences are strong at the group level. So any kind of group level determinations, like adolescents' brains are less developed in areas linked to impulse control and reasoning. That's a very strong inference. So it's a natural fit to class-based legal arguments. which is why a lot of the initial action with neuroscience has been in juveniles, extending the minimum age of execution from 16 to 18, for instance. A lot of that, the justices were swayed by amicus briefs that had a lot of neuroscience showing how 16 and 18-year-old brains were not very well developed. And finally, I think public policy might be the best fit because public policy has to do with populations, right? And neuroinferences are very strong at the population level. So if we want to change the system, for instance, to get seriously mentally ill treated more in hospitals or in outpatient settings instead of prisons, we have to change the system at all sorts of levels. That's done with public policy. And this is where I want to reach out to you guys. I'm strong in neuroscience, but I'm much weaker in public policy. So if any of you are working on public policy projects that can benefit from some neuroscience, please reach out to me. I'd be happy to help. And that's the end of my talk. Thank you.
Thank you, Tavi. That was just fantastic. So
I'm sorry that went so fast, so I hope people were able to keep up with that.
So there have been several questions, so let me just pick from them. One person asked, to what degree is it even possible to assess brain activity retrospectively at the time of the crime when you're using an fMRI?
Yeah, that is such an important question. And that was actually also one of the very sharp observation. So yeah, I mean, for insanity, obviously, we care about the person's mental state around the time of the offense, not years later when you're putting them in the scanner. So that's a really big limitation. So for the Semrau case, he was trying to introduce fMRI evidence that he was telling the truth, but this was being tested, years after the actual time period of the offenses. So that temporal mismatch is a really big objection, and it's an absolutely valid objection to have.
Yeah. And do you want to talk about the impact of neuroscience on juries?
Yeah, so that's where the seductive allure comes in. So there's a lot of really interesting. So if you just Google seductive allure of neuroscience, there's some seminal papers that will come up, and I sometimes I go over those studies because they're so interesting. But they basically demonstrate that when you give people the identical case, and both cases based on nonsense, but you show brain images along with the nonsense, suddenly the nonsense seems much more plausible and persuasive. And I've certainly seen in the courtroom, the fact is there are a lot of really phrenologically based neuro experts out there, experts in air quotes. And if they're not opposed, those arguments can be very persuasive, because I bet you most of you, like going over the studies, had trouble discerning what the problem was, right? And this is UCSF. You guys are all at the top of your games. It's hard to spot these reverse inference errors and skewed population errors in reasoning. So it's really easy to let things slide if you don't have an opposing expert to point them out. So I think the seductive allure is real. I should mention that there have been larger studies to try to demonstrate seductive allure of neuroscience that have failed, basically had a negative result. But my intuition and my experience in the court shows that there's definitely a seductive allure.
Then there's a question about the retest reliability in terms of the stability of fMRI findings over time.
Yeah, and that's been a huge objection. That's actually mentioned in one of the papers I wrote about this. Basically, the studies, most neuroscience studies are small. And yeah, I mean, most fMRI studies, you can't even replicate it it can't be independently replicated. And in fact, even the same lab often has trouble replicating their own fMRI study. The fMRI is a very weak signal. I mean, you're just looking at these tiny changes in blood oxygenation levels, and it's subject to all sorts of artifacts, and it's a highly processed image with many steps. So it's very fragile. So these lie detectors, there are studies that demonstrate that fMRI lie detectors are 99% accurate. In these small lab settings, but they're super fragile. If you do anything, if you use any kind of countermeasure, if you slightly vary the scenario, the lie detector completely breaks. They're completely unrobust. This is actually a general finding. Like at this point, you'd think like you have AIs reading X-rays, for instance. That's a simple example. Give lots of examples and show it the results. But the problem with modern AIs trying to read X-rays is that they're super fragile. If you change the setting on the machine slightly, it totally breaks the AI. So basically, replicability is a problem, and you get around that by just doing lots and lots and lots and lots of replications. But academic labs won't do that because that's not what gets you the Nobel Prize, right? So you really need industry to do it or some other group to do it, but who's going to do it is the issue.
Yeah. Well, it's just about 9:30, so thank you again for a wonderful presentation, and we'll stop here, so thank you.
Thank you very much. And thank you so much for the invitation. Take care, guys.